This video is for first year BSc, first semester, in the subject Food Science and Nutrition, paper Human Physiology, composition of blood, continued in this session also. Hemopoiesis. Hemopoiesis or hematopoiesis is the process of formation of new blood cellular components. As the name itself says, hemo means to blood, poiesis is synthesis. Hematopoiesis and erythropoiesis are two processes which are involved in the formation of mature blood cells. All blood cells are generated from a progenitor cells called hemopoietic stem cells. Hemopoietic stem cells are nothing but undifferentiated cells that have the ability to continuously divide and differentiate into various other kinds of cells or tissues. They have the ability to give rise to new stem cells, or they can self-renew. Stem cell is a source of all kinds of blood cells. Stem cell is a pluripotent and has an ability to develop into all kinds of cells. From all the cells, stem cells is most resistant to damage. The process of division and differentiation of hemopoietic stem cells is highly regulated. Hematopoiesis begins during the fetal life in the yolk sac, and later the liver and spleen. At birth, it occurs in bone marrow. These stem cells can be classified into four broad categories based on their ability to differentiate. Stem cells are of four types. Totipotent stem cells, which are found only in the early embryos. Each cell can form a complete organism. Pluripotent stem cells are obtained from the inner cell mass of the blastocyte. They are able to differentiate into almost all cells of the 3 germ layers, but not into an embryo. The third type is the multipotent stem cell, found in most tissues, produce a limited range of differentiated cell lineages appropriate to their location. The last type, that is fourth type, is unipotent cell, capable of generating only one cell type, epidermal stem cells. They are found in adult liver stem cells. Hemopoietic stem cells are bone marrow cells that are capable of producing all types of cells. They differentiate into one or other type of committed stem cells. They are also called as progenitor cells. These in turn form the various differentiated types of blood cells. Within the red marrow, hemopoietic stem cells, also called as hemocytoblast, divide to produce various blast cells. Each of these cells mature and become particular formed element. The stages of development of stem cells for the production of different blood cells. First, they will be potentially ready to differentiate. They get converted into progenitor cells. These progenitor cells will give rise to precursor cells or blast cells. Then these precursor cells mature to different types of cells: RBCs, WBCs, platelets, and lymphocytes. Progenitor hemopoietic stem cells slowly self-replicate, differentiating into cells that are multipotent but have reduced self-renewal capacity. The progenitor cells for blood cells are commonly called colony forming units because they give rise to colonies of only one cell type. There are about 4 major types of progenitor cells or colony forming units. First one is the erythroid lineage, then thrombocytic lineage, granulocyte monocyte lineage, and lymphoid lineage. Erythropoiesis is the process of making erythrocytes, that is RBCs, begin with the formation of pre-erythroblasts from hemopoietic stem cells over 3 to 5 days. Several stages of development follow as ribosomes proliferate and hemoglobin is synthesized. Leukopoiesis is the process of making leukocytes and is stimulated by various colony stimulating factors, which are hormones produced by mature white blood cells. Hematopoiesis is completed through 5 processes, that is different formation of different cell types: erythropoiesis, lymphopoiesis, granulopoiesis, monopoiesis, and thrombopoiesis. The main difference between hematopoiesis and erythropoiesis is that hematopoiesis is the formation of mature blood cells, all types of cells, whereas erythropoiesis is the formation of red blood cells or erythrocytes. This picture shows the process of hematopoiesis. From the stem cells, that is hemocytoblasts, the lineages are produced: common myeloid progenitor cells and common lymphoid progenitor cells. These progenitor cells, they give rise to megakaryocyte cells. They become thrombocytes, mature thrombocytes. The same myeloid progenitor cell give rise to precursors of RBCs and WBCs. Here, precursor cells of RBCs, they get matured to RBC cells and released into the bloodstream. The other myeloblasts, they give rise to different types of WBCs and basophils, neutrophils, eosinophils, and monocytes are produced as matured cells. On the other side, common lymphoid progenitor cells, they produce or differentiate into natural killer cells such as large granular lymphocytes and small lymphocytes. These small lymphocytes again differentiate into T lymphocytes and B lymphocytes. Where does this development of blood cells occur? At birth or before birth, in the embryonic stage, the hematopoiesis occurs in yolk sac, liver, spleen, thymus, and lymph node of the fetus. After birth, it occurs in bone marrow, especially in long bones of children. In adults, it occurs in axial, skeleton bone marrows. Erythropoiesis, as I already told you, is the process of the production of RBCs from the erythropoietic stem cells to mature blood cells. It is stimulated by decreased O2 production in circulation, which is detected by kidneys, which then secrete the hormone erythropoietin. This picture shows the stages of maturation of RBCs. From the hemocytoblast stem cells, they are very big in size. The nucleus in these cells occupies almost whole of the cell with open chromatin containing 4 to 5 nuclei. They are deep basophilic in nature. Hemoglobin is absent. Mitosis, cell division, will be present at this stage. In the next stage, that is called pre-erythroblast stage, the cell size decreases. The nucleus occupies 3/4 of cell volume. 2 to 3 nucleoli is present. Chromatin is open. Hemoglobin is absent. Active mitosis is under processing. Next stage is early normoblast. The cell size further decreases. No nuclei is found. Chromatin of the cell condenses. Here also hemoglobin will not be present, or it is absent. Active mitosis, cell division, will be occurring in this stage. Next stage is intermediate normoblast stage. The cell size still decreases. The nucleus size also further decreases. Chromatin further condenses. Marked cytoplasm polychromatophilic staining can be seen here. In this stage, intermediate normoblast stage, the hemoglobin starts appearing. Mitotic cell division continues. Late normoblast stage has two stages, early stage and late stage. In the early stage, the cell size further reduces. Nucleus becomes very small with chromatin dot cartwheel appearance. Hemoglobin content increases. The mitosis stops in this stage. In the late normoblast stage, the cell size further decreases. Nucleus degenerates, becomes uniformly deeply stained pycnotic, and hemoglobin appears clearly. Mitotic cell division is absent. The next stage is the production of immature RBCs. They are reticulocytes. The cell size will be 7 to 8 micrometer. No nucleus is seen. Remnant of RNA will be present. Hemoglobin is present in this stage. No mitosis, cell division seen. The last stage is the erythrocyte stage, that is formation of completely matured RBCs of 7.2 to 7.4 micrometer in diameter. No nucleus. The formation of hemoglobin is complete, and this completely matured RBC cells appear in the circulation, or they get released into the circulation. Now we'll see the functions of blood. As we all know that with the help of hemoglobin, blood transports oxygen from lungs to the tissues and carries carbon dioxide from tissues to the lungs for excretion. Along with oxygen, blood helps in the transport of food materials or nutrients to the tissues. Along with that, blood transport metabolic waste products for the excretion sites as kidneys, lungs, skin, intestinal, and for the intestinal removal, etc. The transport metabolites from the tissues to another in association with the kidneys and lungs. Blood maintains acid-base equilibrium of the body. It maintains steady osmotic pressure in the tissues and fluids of the body, being assisted by kidneys and the skin. The plasma proteins assist in the exchange of water in the body from the tissues to the blood and vice versa. It maintains the body temperature at a constant level during its circulation by absorbing and releasing water. It transports hormones from site of production to different tissues. By clotting, it protects body from losing blood. The WBC forms a defense against microorganisms. The next part under this chapter is to learn about coagulation of blood. Blood coagulation or blood clotting is a sequential process that involves the interaction of numerous blood components called coagulation factors. Why blood coagulation is essential? The process of blood clotting and then the subsequent dissolution of the clot following the repair of injured tissue is essential to have the hemostasis. Hemo means blood, stasis is to stop. The loss of blood due to any injury that may be due to accidents or damage to the tissues or blood vessel can lead to the disturbance to the homeostasis. That can lead to dehydration in the body that may lead to many complications. Blood clotting should be prevented to prevent the loss of excess blood from the body in order to maintain the normal internal environment. The clot of the blood is formed at the injured tissues and plugs the injured vessels or open or cut vessels to stop the bleeding. The process of blood coagulation, as I told you, requires many components which are called coagulation factor. There are about 13 principles or factors, blood coagulation factors, which are presented here on the picture. They are fibrinogen, prothrombin, etc. There are about 13 factors which are depicted as Roman letters. Apart from these, coagulation factors are manufactured by the liver. Ionized calcium is available in the blood and from intracellular sources. Phospholipids are prominent components of cellular and platelet membranes. Coagulation in physiology, the process by which a blood clot is formed. The formation of a clot is often referred to as the secondary hemostasis because it forms the second stage of process of arresting the loss of blood from a ruptured vessel. The first stage, primary hemostasis, which is characterized by a blood vessel constriction, that is called vasoconstriction, and platelet aggregation at the site of injury. This picture you can see When a blood vessel is cut or ruptured, the blood oozes out. The blood vessel around the wound get constricted by the substances which are released by the injured tissues or the membrane which are present immediately beneath the blood vessel. Here, to this injured site, the platelets become activated and they try to move towards the injured site and try to stick to the injured place or site. The platelets, they become sticky and clump together to form platelet plug. These platelets and damaged tissue release clotting factors. Here, for example, clotting factor 8. The blood clotting mechanism to form fibrin which acts like a mesh to stop the bleeding. Here, this picture demonstrates the damaged blood vessel activates the cut surface of the blood vessel itself, oozes out the active thromboplastin near the vessel site and vascular system. Platelets aggregate, cause vasoconstriction. Along with these coagulation factors, they help each one to be stimulated to bring this vasoconstriction. Blood components like prothrombin along with the calcium ion which is already present in the blood converts inactive thromboplastin into active thrombin. Active thrombin converts the fibrinogen to active state in presence of calcium, which forms or gives rise to fibrin threads. These fibrin threads cross one another to form a sort of network. You can see that in the photo which is presented here. And into that meshwork of the fibrin threads, the blood cells and platelets are entangled to form a clot. That clot plugs the injured or opened blood vessel to stop the bleeding. This is the process of blood coagulation. Thank you.